Hi everyone, welcome back to my Seurat video tutorials. In this video, I'm going to show you how to use the FindMarkers function. This function can help you find the markers to identify the cell types in a cell cluster. In reality, people actually use this function more to find differentially expressed genes across different experimental conditions. So in my previous tutorials, we integrated 3 single-cell RNA sequencing dataset for normal human lung tissue. So we can load this integrated dataset into R. For this demonstration, we can use very low resolution to reduce the cluster numbers. We can use 0.01 to find the clusters. Now we can run the dim plot to find out how many clusters we have. Let's zoom in. You can see with the resolution 0.01 we have 7 clusters. So normally you should know some marker genes for most of the cell types in your tissue if you have some experience in your research field. For example, in human lung tissue, we use 4 markers to identify epithelial cells, endothelial cells, mesenchymal cells, and immune cells. EpCAM is a marker for epithelial cells, and CLDN5 is a marker for endothelial cells. Collagen 1A2 we use it as mesenchymal cells, and PRC is a general marker for immune cells. We can use the feature plot to see their expression in this dataset. Let's run the feature plot. So we can zoom in to look at their expression in different cell clusters. You can see EpCAM is highly expressed in those cells, so they are epithelial cells, and the CLDN5 are expressed in this cell cluster, so they are endothelial cells. Collagen 1A2 are expressed in these 3 clusters, so they are mesenchymal cells. Cells express the PTRPC, so we know they are immune cells. So in most human tissue, you will have the cell population for endothelial cells, mesenchymal cells, and immune cells. In the lung tissue here, the epithelial cells are specific cells for the lung tissue. If you are working in different tissue, for example, in the nervous system, you still got like here endothelial cells, mesenchymal cells, immune cells, but for the nervous system, you will have nervous system specific cells. Probably have the neurons if you work in the central nervous system. For the peripheral nervous system, you have like Schwann cells. So we know the mark gene expression for epithelial cells, endothelial cells, mesenchymal cells, and immune cells. So let's have a look at the dim plot again. We can zoom in. Now you can see we know cluster 2 are epithelial cells, cluster 1 are endothelial cells, and cluster 4 are mesenchymal cells, and cluster 0, 3 they are immune cells. Now we can look at the function find the markers. So when we integrate the 3 dataset, we set the default array as integrated. Now we need to use the FindMarkers function, we need to set the default array as RNA. Let's run this function. So now we can use the FindMarkers function to find the markers for each cluster. So first, let's have a look at the arguments for the FindMarkers function. So here is the name of your Seurat object. Ident1 tells this function that we want to identify markers for which cluster. In this case, we are identify markers for cluster 2. So the minimum percentage argument requires a gene to be detected at the minimum percentage in a cell cluster. So here we set at 50%. So by default, the FindMarkers function identifies both positive and negative markers for a cell cluster. If we just want to return positive markers, we can set positive only as true. So the last argument is log_fc threshold. The log_fc threshold argument requires a gene to be differentially expressed by some amount between cluster 2 and the rest of the cell clusters. So we can run this function to find the markers for cluster 2. So if you want to find markers for all the cell clusters, you can run the FindMarkers function one by one for each cluster. But Seurat has another function called find all marks. So this function will find the markers for all cell clusters in your dataset. So the difference for find all markers is we don't need to set identity for cell clusters. Now let's have a look at the markers for cluster 2. So we can reorder it according to the order of average log 2 FC. So you can see the highest log FC is SFTPC TPB. So we know those markers are type 2 epithelial cells. And for example, the AGER is a marker for type 1 epithelial cells, and SCGB3A1 is a mark for cluster cells. So those marks indicate that cluster 2 is a cell cluster for epithelial cells in the lung tissue. So when we use the feature plot function, we showed you that cluster 2 expressed the EPCAM. So we know cluster 2 is a cluster for epithelial cells. So Seurat has several functions to visualize mark gene expression, such as feature plot, dot plot, and violin plot. So we can put the gene names identified in cluster 2 and have a look at their expression. So we can run the feature plot. Let's zoom in. You can see those markers are highly expressed in cluster 2. It's very low in the rest of the clusters. We can run a dot plot. Let's zoom in again. Again, you can see those markers are highly expressed in cluster 2. So the dot size represent the percentage of cells that express this gene in cluster 2. Let's run the wiring plot again. So again, you can see those genes are highly expressed in cluster 2. So this just different ways to visualize their expression in cluster 2. So as I mentioned in the beginning, the FindMarkers function actually identify differentially expressed genes. So the FindMarkers function is a very useful function to study differential gene expression and gene-related signaling pathways in specific cell types. So to test for differential gene expression between two specific cell groups, for example, we are going to test the cluster 0 and the cluster 3. So we know both are immune cells. Through differential gene expression, we can identify what kind of immune cells they are. So to do so, we just need to specify the identity 1 as the cluster 0, and we have identity 2 here as the cluster 3. So let's run this function. So we identified the differentially expressed gene between cluster 0 and cluster 3. So let's have a look at the differentially expressed genes between cluster 0 and cluster 3. So here is the data frame for the result. In the first column, it's the gene names. The second column is the unadjusted p-value. The last column is adjusted p-value. In the average log2fc column, it shows the log-fold changes of the average expression between the two groups for those genes. So positive value indicate that this gene, for example S100A8, is highly expressed in the cluster 0. Negative value means no expression in cluster 0 and high expression in cluster 3. So the percentage 1 show the gene expression percentage in cluster 0, then percentage 2 show the gene expression percentage in cluster 3. So you can see we can use the FindMarkers function to identify differentially expressed genes between different cell clusters, and later we can use this differentially expressed gene to analyze what kind of signaling pathways are activated in the cell cluster. So if you want to compare one cell cluster against two, you can name two cell clusters in identity 2. You can see here we set identity 1 as cluster 0 and identity 2 as cluster 3 and 6. So we can run this function. So this function will find the differentially expressed gene between group 0, 3, and 6. So in summary, in today's video, I show you how to use the FindMarkers function, and also showed you the FindMarkers function is a very useful function to identify differentially expressed gene between different cell groups. So I'm going to stop here today. So please subscribe my channel if you would like to watch my upcoming video tutorials.